Today we're going to talk about muscle. Contractivity is a fundamental property of protoplasm. Most all cells contract somewhat, but muscle cells have especially adapted to convert chemical energy into mechanical work. Today we want to identify the different types of muscles, a morphologic basis for the different types of muscle, and then distinguish the modes of excitation for the different types of muscle. There's three basic types of muscle. We have smooth muscle, which we have in a GI tract, cardiac muscle that we have in a heart, and then there's skeletal muscle that we have to move our different components of the body. If we look at these different types of muscle, we see the smooth muscle is like a cigar shape. It's a small cell, and the nucleus is in the center of the cell. So in cross section you see the center of the cell. It's very characteristic of smooth muscle. In the GI tract, you usually see some longitudinal and cross sections of the same smooth muscle cells. In the duodenum, we can see those. This is the muscularis externa, and that's where we see this smooth muscle. It's inner circular and outer longitudinal muscle that we can see there. If we look at this toluidine in blue, again we can see the muscle layer that goes from the outside in through there. And if you look right in close, you can see this little line in through there. That's also muscle right in through here, small smooth muscle above the submucosa. This is submucosa muscularis externa, and this is some nerve cells that regulate that muscle. Muscularis externa, muscularis mucosa is what we're seeing. Again, we can see the muscularis externa, submucosa, and then the muscularis mucosa right in through there. The muscularis mucosa sends projections of cells in there to move the villi. We can see some of the smooth muscle cells running through here. There's another one right there, which come from the muscularis mucosa. A higher mag of that, we see the smooth muscle cells of the muscularis mucosa. We also see the muscularis externa. We have intercircular outer longitudinal muscle, and we can see the iron box plexus. These are nerve plexus cell bodies that regulate the contraction of Paracelsus that goes on here. Also in the submucosa we can see nerve cell. This is your iron box plexus, which regulates the contraction of the smooth muscle cells in the muscularis mucosa. On this slide we can see the muscle layers again, smooth muscle layers that we see. This is blood in through there. This is a little venial, but these are the smooth muscle cells, some cut longitudinally and some in cross section. Characteristic of smooth muscle is that individual cells, you can see individual nuclei, whenever they're in a partly contracted state, the nucleus is corkscrew in shape. So instead of look like a cigar, it looks kind of like a corkscrew. In this slide 475, we can see the corkscrew shaped nuclei of the smooth muscle cells, and this happens to be uterus. Another characteristic of smooth muscle is it has a basal lamina. So it has like a basement membrane around each of these, and that is PAS positive. So the nucleus is in the center. Nucleus kind of shaped like a cigar, except whenever it's a corkscrew, and the cytoplasm on the outside has a PAS positive basement membrane like epithelium. And here we can see in this slide a little bit of pink running through here, which is the basement membrane of the cells. In this particular one, we're seeing the ductus deferens which has the luminal opening in through there. So this is epithelium, and all this is smooth muscle. So we have an inner and outer longitudinal muscle and center circular muscle layers. That is, it's all one muscle, but it's all smooth muscle, inner circular, outer circular, and inner. Longitudinal and circular. This is a cross section of this, so it's inner longitudinal, outer longitudinal, and middle circular muscle. Also, we see smooth muscle in this little artery here and little venule that we see there, and we see some. This is nerve in through there that we're seeing, but here we see the longitudinal and cross sections of smooth muscle. This is a H&E again, showing you all the different smooth muscle cells in through their epithelium on that surface, a blood vessel, but these are smooth muscle cells around this blood vessel as well. In the uterus, there's a host of smooth muscle cell bundles that you see there. These are cross sections of the bundles, showing you that there's a host of cells in those bundles. Here we see smooth muscle cells again. And this is a uterus. This is a myometrium of a uterus. In the oviduct, we can see smooth muscle cells running through there. Also, the smooth muscle cells you see right there in the blood vessels as well. If we look at a blood vessel, here's the lumen of the blood vessel with an electron microscope. We see the endothelial cell, and we see the internal elastic lamina, and then we see the smooth muscle cells. Smooth muscle cells are characteristic by a lot of filaments because they do have actin and myosin. Also, they have these fusiform densities, these densities that you mostly see on the surface, but not always on the surface. And these are where filaments are attached to one another inside the cell and to regulate the contraction of the cell. So this is one smooth muscle cell, there's another smooth muscle cell, and another smooth muscle cell. And if you look real closely, we can't at this magnification, but you should see a basal lamina because there's a basement membrane that's associated with it. Here we can see the basal lamina of the smooth muscle cell. This is the endothelial cell here. There's a lumen that we see there. Another characteristic of these cells, it has these apical caveoli, and these like little pinching vesicles that pinch off from the surface that you see around, or maybe they're invaginations to take up calcium. Now, here we see some smooth muscle cells with diffusiform densities and the innervation that they can get. So this is an axon that's synapting on these cells to regulate their contraction. There's another one, so this will be a swan cell cytoplasm, axon, and this is mitochondria in there, but these are the fusiform densities of the smooth muscle cell. In the esophagus, we have more smooth muscle cells in through there, and here we see a combination of smooth muscle cells, these smaller cells, and skeletal muscle cells. Skeletal muscle cell does not have a nucleus in the center, it has a nucleus on the periphery. And also it's multinucleated cell, where smooth muscle cell, individual small cells, the skeletal muscle is a series of nuclei, multinucleated cell with the nuclei on the periphery. And if you look at more closely at skeletal muscle, you can see there's a striation. You do not see striation in smooth muscle, hence smooth, but you do see have sets of striations that you see in longitudinal view of skeletal and cardiac muscle. This is the nucleus of the muscle, and you can see the different components which make up the striations. You have the A band, which is a dark band, the I band, which is a light band. The Z line is in the middle of the I band. There's also another band, the H band, that's in the middle of the A band as well. Z line, H band, I band, A band, the different bands that we see there, and each of these is a sarcomere. From one Z line to another Z line is one sarcomere, and that's the contracting unit, functional and structural unit of contraction of the skeletal muscle cells. And here we can see what happens when you have contraction to occur. It brings in the Z lines close to one another, closer. Whenever it's relaxed or stretched, the Z lines are further apart. And so the actin filaments move in relation to the myosin. Myosin stays put. The actin with the Z line is what changes there. And here we can see some hypercontracted. That's where the A band would be, and the I band would be there too. There's your Z line. The Z line's in the middle, of course. In this case, you can hardly see the A and I band because it's so contracted, and so one sarcomere is from here to there, another one from there to there. And remember, these different bundles of fibers are organized by intermediate filaments. So we see that again in this view, the striations that we can see in these, and sometimes in the muscle you'll have a muscle spindle where you actually have some of the fibers inside a capsule, and this is responsive to stretch, and so it tells the muscle don't stretch anymore, or how it's being stretched. Here we see skeletal muscle and tongue. We can see the fibers there, the nuclei on the periphery of these muscle cells, and so you can move your tongue back and forth. It's under your control. And here we see a hieromag. You can see the nuclei on the periphery around one muscle fiber, and then here you can see them cut in a longitudinal view, which will allow you to see the striations. The dark band is the A band, the I band is in between. And the A and I band stand for isotrophic or anisotrophic, and how it responded, polarized light responded to it. You can see the striations that we see there. Again, the skeletal muscle has a nuclei on the periphery, and it's multinucleated cells. Here we see some young skeletal muscle cells. It looks a little bit like smooth muscle because the cells are not very large, but the nuclei are still on the periphery, as we can see. This is one of those muscle spindles where you actually have some fibers of the muscle fibers within a capsule that controls or detects stretch. So we can actually see some striations sometimes in the muscles cells which are inside the capsule of the muscle spindle. The other type of muscle is striated, and that's cardiac muscle. And the cardiac muscle has an A and I band, just like skeletal muscle, as you can see here. But also it has a heavy band too. These are intercalated discs. That is, this is where one cell attaches to another cell, and it's always on the Z line. So it's the Z line is where the cells attach. And here we see the heart, and this is a cardiac muscle cell. Show you the transverse tubule that goes through there, the transverse tubule here and here. Transverse tubule is on the Z line. So here again we have the A band, which is the dark band, and the lighter band, which is the I band, and the Z line is in the middle of that band. In addition to these bands, we have this heavy band here, which is an intercalated disc. And if you look real good, you can see gap junctions between adjacent cells. That's how one cell tells another cell that it's contracting. Here we see some cardiac cells in a blood vessel, not the heart, but the ones coming into the lungs. You see the A band and the I band. You could even see the Z line on these. Now, cardiac muscle has the nucleus in the center. So it's larger than smooth muscle, but not as big as skeletal muscle, and the nucleus is pretty much in the center. So here we can see these are cardiac muscle that we see through there, striated muscle, and it has an intercalated disc. So here we can see the striations, and these heavier discs here are here we can see it very nicely, the intercalated disc where one cell is attached to another cell. Also in the heart we see some lipofuscin. Lipofuscin is characteristic of old heart. In slide 125 we can see the intercalated disc very nicely and the sarcomeres within. So here we see the lipofuscin, which remembers is the undigested lysosomal activity. And here we see them in the cardiac muscle as well. So in the cardiac muscle we see some lipofuscin, we see the nucleus in the center. It's not a multinucleated cells. It may have two nuclei, but that's pretty much in, and it has the intercalated disc, which is where the cells are attached to one another. Again, we can see cardiac cells, the A and I band, and these long things here are mitochondria located in there, and this is in the lungs itself. So cardiac muscle, as we see the intercalated disc where cells are attached, the A and I band, the typical of cardiac muscle cells and skeletal muscle cells. Here we see some cardiac cells with a nucleus in the center versus young skeletal muscle cells, and the nuclei are located on the periphery. Even though it's about the same size, because this is fetal tissue, the nucleus is still located on the periphery as opposed to in the center of cardiac muscle cells. This is, again, skeletal muscle cells that we see. Here we see some midterm practical exam questions that we have here. And you can see just some of the questions. What are two functions of this organelle? This organelle happens to be mitochondria, so to produce ATP, calcium storage, cholesterol, side chain cleavage is some of those things. Here's another one, number 17. What's the composition of this structure and what is its function? It has to do with the fibrous lamina. It's the felt-like intermediate filaments inside the nuclear envelope. It supports the nuclear envelope, supports nuclear pores, and keeps them separate from the chromatin. Here's another question. What are two major components of extracellular matrix ground substance in which these structures are embedded? And those are structures which we identify with type 1 collagen. So you got hyaluronic acid and glycosaminoglycans, or proteoglycans is another thing. Classify this vessel. It was a muscular artery. Name this cavity or give its role in remodeling. It's the absorption cavity whenever a bone is being remodeled. What role do these cells play in autoregulation of microvascular smooth muscle cells? When oxygen tension is low, they relax. When they relax, the blood vessels expands and more blood comes through. What does a high density percentage of white blood cells of these cells indicate? In the bone marrow, band stab cell, that means there is infection as a lot of neutrophils are needed. Giving a nuclear or cytoplasmic feature of these cells in bone marrow that distinguish it from the other developing steps. Okay, this is in bone marrow, and it's a spherical nucleus, thin rim of deep blue cytoplasm. So this must be, if it's a spherical nucleus, it must be then the red blood cell series. So if it's non-fractured face, then it would be a pronormoblast. What is the function of this encapsulated structure, which is a muscle spindle. It's a muscle stretch receptor. Okay, so these are some of the things that you might consider. So today we looked at muscle, identify the different types, the morphologic basis for the different types and where they're found in different places in the body, and distinguishes the different types of modes of excitation that goes on in muscle. Thank you.